Hello students, welcome to EPG Pathshala. I am Dr. Vishwanath Tiwari from Central University of Rajasthan. Today I am going to discuss the module that is mechanism and kinetics of non-competitive as well as uncompetitive inhibition. This module has been taken from paper Molecular Enzymology and Protein Engineering. In this module, we will discuss about mechanism and kinetics of non-competitive inhibition, how it differ from uncompetitive inhibition. At the same time, we will also discuss the examples of different types of non-competitive inhibition or uncompetitive inhibition. We will also discuss how we can determine the dissociation constant of non-competitive inhibitors and mechanism and kinetics of uncompetitive inhibition. So, enzyme inhibitor is either irreversible, reversible, feedback or suicide. But in this module, we are discussing about reversible inhibition. So, when we are talking about non-competitive and uncompetitive type of reversible inhibition, so they all involve weak non-covalent interaction such as hydrogen bonding, hydrophobic interaction, van der Waals forces and salt bridges. They all are involved in the stability of non-covalent interaction that playing role when a non-covalent inhibitor bind to the enzyme or uncompetitive inhibitor bind to the enzyme. It seems that they are very weak interactions, but if you are taking cumulative effect of these interaction, then they result into strong interaction. So, based on different type of interactions, reversible inhibitor are of different types, but in this module we are discussing only about non-competitive inhibition and uncompetitive inhibition. So, based on effect of varying concentration of enzymes, substrate and inhibitor, reversible inhibition is either competitive, either non-competitive or uncompetitive, and a special case of mixed inhibition. But in the non-competitive and uncompetitive, we will discuss it in the detail in this module. So, in non-competitive and uncompetitive inhibition, in contrast to the competitive inhibition, here we do not have any structural similarity between substrate and inhibitor. So, due to the absence of structural similarity between substrate and these inhibitor, they are rarely used in the drug designing. So, the study of non-competitive inhibition and uncompetitive inhibition have less significant in drug designing, but they are very important when you are talking about study of various biological reaction, because in various biological reaction we are going to study different inhibitors which is based on non-competitive inhibition or uncompetitive inhibition. So, therefore, in this module we are discussing about characteristic, kinetics, mechanism, examples of non-competitive and uncompetitive inhibitor. The mechanism of non-competitive inhibitors is based on non-structural similarity between substrate and non-competitive inhibitor. That means that your non-competitive inhibitor doesn't have any structural resemblance with the substrate. Therefore, there is no competition for the binding of non-competitive inhibitor with the enzyme. So, non-competitive inhibitor can bind to the free enzyme or it can bind to the enzyme substrate complex. But if your inhibitor is bind with the free enzyme, then EI complex is formed. So, substrate can also bind to the enzyme which is free as well as to enzyme inhibitor complex. So therefore you can say that the binding of non-competitive inhibitor as well as substrate are different and independent. They are not affecting the binding of each other. It is interesting to note that non-competitive inhibitor binds to the enzyme site other than the active site, and it is the allosteric site that is well known other than the active site in the enzyme. But it is important to mention here that the non-competitive inhibitor most of the time bind to other site, but that is not denoted as allosteric site because they will not influence the binding of the substrate, because allosteric site is a one that influence the binding of substrate to the remaining active sites. Therefore, except mixed inhibitor where the binding of inhibitor influence the binding of substrate, all the non-competitive inhibitor binding sites are non-allosterics. The resulting enzyme substrate inhibitor complex is catalytically inactive because non-competitive inhibitor prevents the proper positioning of catalytic centers or catalytic residues that is important for the converting substrate into the product. Therefore, ESI can be considered as an inhibitor modified ES complex which is unable to form product. This shows that the formation of ES complex is not altered whether you have a non-competitive inhibitor or you don't have, but it is the product formation that will alter. So if the ES complex formation is not altered, KM will not alter. But because of the formation of ESI complex, this ESI complex or modified ES complex with the inhibitor, they will not form any product. So Vmax will decreases. In this figure, you can see that we have two different sites. One for binding of substrate, that is active site. Other is a binding of inhibitor, that is a inhibitor binding site, and we have an enzyme. So when you can see that from left side, when there is a possibility that inhibitor will bind before the binding of substrate, so inhibitor will bind, that is followed by binding of substrate. And you can see from the right side, the substrate can bind, that will followed by the binding of inhibitor. So we have two type of, even three type of complexes here. From right side you can see enzyme substrate complex is there. If from left side you can see enzyme inhibitor complex is there, and we have one more, that is enzyme substrate inhibitor complex. So if you see towards left side, then ESI complex is there which never form any product. But if you see from right side, that enzyme which is having substrate and it is existing as a ES complex, they will form product. So the completion of the reaction in the presence of non-competitive inhibitor is depend on the enzyme undergoes conformation changes that is blocked by the inhibitor. So the which conformation changes which is blocked by inhibitor is regained by any means, then you can reverse the process of inhibition here. The kinetics of non-competitive inhibition. Non-competitive inhibition or non-competitive inhibitor will bind to free enzyme, it can bind to ES complex and inhibit the formation of the product by forming ESI complex. At any concentration of inhibitor, few ESI complex will always exist, and this ESI complex is non-productive. Therefore, Vmax will always decrease in the case of non-competitive inhibition. Decrease in the Vmax does not means that inhibitor has decreased the rate constant of breakdown of ES complex to product, but it is the equilibrium of ES complex that is decreased due to the formation of ESI complex. Because if you have ES complex, that is converted into the product, but if few ES complex is trapped along with the inhibitor by producing ESI complex, they will never form the products. So Vmax always decreases in non-competitive inhibition. So the Km, which is measured how a substrate is interacting with enzyme. If you remember, the non-competitive inhibitor will bind to other site where your substrate will bind. So substrate will bind to the active site and non-competitive inhibitor will bind to other site. Therefore, the Km remain unchanged in the presence of uncompetitive inhibition, because they will only bind to free enzyme or ES complex, but they will never affect the binding of substrate to the active site. The Michaelis-Menten equations and plot can also be derived for non-competitive inhibitor. You can see enzyme combined with the substrate producing ES complex. Enzyme can combine with the inhibitor producing EI complex. If ES complex is there, it will bind to the inhibitor. If EI complex is there, it will bind to the substrate, and finally it will produce ESI complex. And the ES complex which does not combine with the inhibitor is going to form products. So if you derive Michaelis-Menten equations for non-competitive inhibitor, then you can see that the equation is look like V0 is equal to Vmax into substrate concentration divided by 1 plus I upon Ki. In the bracket you have Km plus S. Here if you see this equation, it seems that we are multiplying Km plus S by 1 plus I upon Ki. But please remember this, we are putting 1 plus I upon Ki in the denominator not to show increase in the Km and substrate concentration, but here we are putting it in the denominator to show there is a decrease in the Vmax, because it is a general rule when the Vmax is, if any kinetic parameter is decrease, you are going to divide that with the factor of decrease. When there is an increase in the kinetic parameter, you are multiplying it with the factor of the decrease or increase. In this equation, V0 is initial velocity, Vmax is a maximum velocity, and Km is Michaelis-Menten constant, I is inhibitor concentration, and S is substrate concentration, and Ki is a dissociation constant of non-competitive inhibitor. So in this equation you can see that Vmax is decreased by 1 plus I upon Ki. That means we have modification in the Vmax. Therefore, the modified Vmax in the presence of non-competitive inhibitor is known as apparent Vmax, and apparent Vmax is equals to Vmax divided by 1 plus I upon Ki, and this is the factor which is responsible for non-competitive inhibition. So this factor Vmax 1 plus I upon Ki is considered as a inhibitor dependent statistical factor describing the distribution of enzyme substrate complex between ES and ESI form, and it is also represented by alpha. If you see the plot of Michaelis-Menten equation here and you are comparing the competitive inhibitor presence and its absence, you can see that in normal condition we will have the Km which is exactly same as the Km which is present in the presence of inhibitor. So Km will not alter, but if you compare the Vmax of enzyme in absence of inhibitor and presence of inhibitor, then you will find that there is a decrease in the Vmax. As you know that ESI complex is formed which is never forming any product, and that is responsible for this decrease in the Vmax. Similarly, Lineweaver-Brock equation can also be derived for non-competitive inhibition. And if you see this equation, it look like 1 by V0 is equal to Km upon Vmax multiplied by 1 plus I upon Ki into 1 upon S plus 1 by Vmax multiplying by 1 into 1 plus I upon Ki. So, based on Lineweaver equation, Lineweaver plot can also be drawn, and you can see the Lineweaver plot for non-competitive inhibition. As you can see from the graph, it shows that Km either in the presence of inhibitor, either in the absence of inhibitor is remain same, while 1 by Vmax increases in presence of inhibitor. Because this Lineweaver plot is a reciprocal plot, so if 1 by Vmax increases, then Vmax decreases. This is what we are trying to say, is that a particular pattern of graph of non-competitive inhibitor is observed here that is totally different from competitive inhibitors and uncompetitive inhibitor, and that is very useful when you are going to determine the type of mechanism that is particular inhibitor is going to have. So, what we are going to do, we are performing two independent kinetic experiment. In one we have inhibitor and in other we do not have inhibitor, and a particular pattern of Lineweaver graph observed, and that observed pattern will tell you what is mode of your inhibitor. Based on non-competitive inhibition, we can also derive the dissociation constant for non-competitive inhibitor. What we are going to do here is, we are performing two experiments, one in the presence of inhibitor, another in the absence of inhibitor, and Ki dissociation constant can be calculated with the help of equation where 1 by Vmax apparent is equals to 1 by Vmax multiplied by 1 plus I upon Ki. In this way, when we have two plots, we can see the y-axis intercept. From that, we can determine what is the 1 by Vmax in the presence of inhibitor, and from normal graph we will know what is Vmax of enzyme in absence of inhibitor. And if you determine both and you know the concentration of inhibitor, you can determine the dissociation constant of inhibitor that works with non-competitive mechanism. Not only that, it is a well known that the dissociation constant of inhibitor, that is Ki, is inversely proportional to the binding and affinity of non-competitive inhibitor and enzyme. So, if the concentration of inhibitor increases, then slope of the plot increases and intercept at 1 by V0 axis moves towards higher side. So, apparent Vmax continuously increases. After discussion of kinetics and mechanism of non-competitive inhibition, let us have discussion of different examples that follows non-competitive inhibition. The first example is alanine, which is a non-competitive inhibitor of pyruvate kinase. Pyruvate kinase involves in the conversion of phosphoenol pyruvate into the pyruvic acid. So, alanine is a competitive inhibitor of pyruvate kinase. The second one is silver ion. It reacts with the sulfur hydryl group of cysteine in the polypeptide backbone. So, if any enzyme that uses cysteine for the catalysis, then it will be inhibited by silver ion. This is the one reason why silver nanomaterials are going to be developed as one of the drugs which act non-competitively. The third example is potassium cyanide. It is well known inhibitor which act on the complex 4 of electron transport system, that is cytochrome oxidase, but the mechanism of action of potassium cyanide is non-competitive because cytochrome oxidase is important for the electron transport system and ATP synthesis. The fourth one of non-competitive inhibitor is nifedipine, which inhibits cytochrome P450, and the mechanism of inhibition is non-competitive. Michaelis-Menten equation can also be derived for uncompetitive inhibition. As you know that uncompetitive inhibitor decreases Vmax, it also decreases Km. So you can see in the equation the V0 is equal to Vmax into S divided by Km plus S multiplied by 1 plus I upon Ki. The important point you are going to note here is substrate concentration is multiplied by 1 plus I upon Ki, but Km is not multiplied by 1 plus I upon Ki. Why it so? Because you know that Vmax decreases here, so we are dividing Vmax into S by 1 plus I upon Ki. But we also know that Km also decreases. So we are dividing Km, which is present in the denominator, by 1 plus I upon Ki. So that result into the two things. Because to decrease Vmax, we are multiplying all the denominator with 1 plus I upon Ki. To decrease Km, we are multiplying Km with 1 plus I upon Ki because we want to decrease Vmax, but here because decreasing the Km, we are dividing that Km with 1 plus I upon Ki. So both get cancelled. So Km divided by 1 plus I upon Ki multiplied by 1 plus I upon Ki both get cancelled. So we will only left substrate concentration, that is multiplied by 1 plus I upon Ki. So if you see this equation, it look like Vmax and Km is unaltered, but that is wrong. They are altered, but both they are cancelled because of the solving when you are solving the equation. And this equation, V0 is initial velocity, Vmax is a maximum velocity, Km is a Michaelis-Menten constant, I is an inhibitor concentration, S is a substrate concentration, and Ki is a dissociation constant of inhibitor with enzyme. As you know that in uncompetitive inhibition, we have decrease in the Vmax, we have decrease in the Km. So we will have apparent Vmax, we have apparent Km. So apparent Vmax, as you know that whenever any kinetic parameter decreases, we are dividing that kinetic parameter with 1 plus I upon Ki. If any kinetic parameter increases, we are multiplying the kinetic parameter with the same factor. But here both are decreasing, so apparent Vmax is equals to Vmax divided by 1 plus I upon Ki, and apparent Km, which is also decreasing, is equals to Km 1 divided by 1 plus I upon Ki. Similarly, we can determine the Line-Weaver equation and Line-Weaver plot. So Line-Weaver equation for uncompetitive inhibition can be derived from Michaelis-Menten equation, and it is written as 1 by V0 equals to Km divided by Vmax into 1 divided by S plus 1 by Vmax, that is multiplied by 1 plus I upon Ki. If you see the equation and you are making it equals to the straight line equation, then you can see that there is no change in the slope. That is, if you remember straight line equation, that is y is equal to mx plus c, where y is 1 by v0, m is km divided by vmax, x is 1 by s, and 1 by vmax is the intercept at y-axis. So if you can see that m, which is slope, is exactly same here in presence of uncompetitive inhibitor as you can see in absence of uncompetitive inhibitor. So it shows that your slope is not altered. So what altered here is only the intercept at y-axis, which is c. But when you calculate the intercept at x-axis, then you will find 1 by km will also alter. This calculation of 1 by km has been elaborately discussed in other module of determination of kinetic parameters. So we will find that in presence of uncompetitive inhibitor, there is a change in the Vmax as well as change in the Km. So from the graph you can see that in the presence of uncompetitive inhibitor, 1 by Vmax increases and 1 by Km also increases, which shows that in normal condition, if you reverse the value of 1 by Vmax, it shows that Vmax decreases. If you reverse the value of 1 by Km, it also shows that Km will also decrease. So in the presence of uncompetitive inhibitor, both Km as well as Vmax decreases. After discussion of mechanism and kinetics of uncompetitive inhibition, let's discuss the examples of uncompetitive inhibition. We have 3-4 examples of it. The first one is glyphosate, which is a pesticide. The second one is propionic acid, which is a inhibitor of a, a potent HIV inhibitor converting enzyme, and third one is phenylalanine, which is inhibitor of rat intestinal alkaline phosphatase. Now we will discuss different uncompetitive inhibitors in the details. The first uncompetitive inhibitor, that is glyphosate, which is also known as Roundup. This is a pesticide which is chemically N-phosphonamethylglycine. This is an uncompetitive inhibitor for enzyme that is known as 3-phosphocyclamate 1-carboxyvinyl transferase. This is enzyme involved in the aromatic amino acid biosynthesis. So if you are inhibiting aromatic amino acid biosynthesis by using glyphosate, that will inhibit the growth of particular survival of a particular pathogen. But it is interesting that because of the transgenic productions, we have the production of glyphosate resistant plants. As I have told you that this is a pesticide, so it will kill the pest, but at the same time it also kills the herbs that is there when we are using that pesticides. So glyphosate and Roundup resistant plants are generated where this 3-phosphocyclamate 1-carboxyvinyl transferase activity is replaced by activities of other enzyme. So therefore, in the presence of glyphosate, the pest will be killed, but your herbs will remain as such because they have alternate mechanism, alternate pathways for the production of aromatic amino acids. The second example of uncompetitive inhibitor is protagonist. So we have an enzyme that is known as glucuronyl transferase, which is involved in the glucuronidation of zidovudine. This drug is used as a HIV inhibitor. So this propionic acid is an uncompetitive inhibitor of glucuronyl transferase. Therefore, they are inhibiting the glucuronidation of zidovudine. The third example is L-phenylalanine. L-phenylalanine is an uncompetitive inhibitor of rat intestinal alkaline phosphatase. Although uncompetitive inhibition is not a very common type of inhibition, it rarely happens, but it is an important mechanism, and it is assumed that uncompetitive inhibition is an modified non-competitive inhibition where your uncompetitive inhibitor is dependent on substrate for its binding, while non-competitive inhibitor are totally independent to the substrate for its binding. So if you have non-competitive inhibition, that can classified into two in terms of their modification. The one where the binding of uncompetitive inhibitor is dependent on binding of substrate. Another example of mixed inhibition where the binding of inhibitor is dependent on binding of substrate, at the same time binding of substrate is also dependent on binding of inhibitor. So therefore, a non-competitive inhibition where both are independently bind, in uncompetitive inhibition, inhibitor is dependent on the substrate, and in the mixed inhibition, both are dependent to each other. So students, let's have summarize what we have discussed in this module. In present module, we have discussed mechanism of non-competitive inhibition and uncompetitive inhibition, and we have also discussed what is difference in non-competitive inhibition and uncompetitive inhibition. We have discussed that a non-competitive inhibitor can binds to free enzyme, it can bind to ES complex, but uncompetitive inhibitor always binds to the ES complex. We have also discussed how the kinetics of non-competitive inhibitor is different from uncompetitive inhibitor. We have discussed about different examples of uncompetitive inhibition, different examples of non-competitive inhibition. We have discussed the Lineweaver-Brock plot, Michaelis-Menten equations and plot for these two inhibition. We have also discussed how Lineweaver-Brock plot is used to calculate dissociation constant of non-competitive inhibitor as well as uncompetitive inhibitors. We have discussed that in presence of non-competitive inhibitors, there is a decrease in the Vmax because of the formation of ESI complex. We have also discussed there is no effect of non-competitive inhibitor on the Km of the enzyme because non-competitive inhibitor does not alter the formation of ES complex because both have different binding sites. Similarly, we have also discussed about inhibition of uncompetitive mechanism where we have discussed uncompetitive inhibitor decreases the Vmax as well as Km. We have discussed why an uncompetitive inhibitor decreases the Km. This is because shifting of equilibrium. You know that when enzyme combined with the substrate produces ES complex and uncompetitive inhibitor binds to the ES complex. So therefore, the equilibrium will shift toward the formation of more ES complex, therefore the Km decreases. Uncompetitive inhibitor are similar to non-competitive inhibitor in the mechanism that both doesn't have any structural similarity with substrate. So in this way we understand why non-competitive inhibitor and uncompetitive inhibitor are important. Although they are not important when we are talking about the drug designing, but they are more important when we are talking about the study of biological functions and searching an alternate molecule to regulate biological functions. Thank you.